and translation system. So you can get the protein product in the tube itself, if that is the purpose. But otherwise, the gene alone without any assistance or the system for transcription and translation is just a piece of DNA.
Yeah, I think Deepak is back.
Deepak is back. Good enough.
I am back.
Go ahead, Deepak. We were holding the fort till you come back.
Okay, thank you, sir. Thank you. So I hope, sir, my voice is audible now.
Now it is better. Yes, no problem.
Okay, so sorry for inconvenience caused, sir. I'm really sorry. So I'll proceed from here. So I mentioned in my continue from where I left actually. So we have this sequence with us. This green is basically green and red are basically two strands of DNA. So if we have a sequence, say for instance, for one strand, in this case, I am mentioning here this green strand. If I have a sequence, so I can design a primer from this sequence. So for designing the primer, what we essentially do is we take this region. So I have taken here, you might be able to see that this, if say in this case, we want to design primer of 5 bases. So we can take exactly the forward sequence as such, the like 5 prime C, T, C, A, G. And from reverse end, we can take it at T, C, A, A, T. So now with these two primer, see in the next this you can see I have shown their binding also, how they will bind. So since this forward primer is having C, T, C, A, G, so it will bind to the complementary sequence which is present in the another strand, the complementary strand, and this TCAAT will bind like this. So in this way, you can go and amplify this particular region. So exactly what is happening here, I told you, in denaturation, DNA is being denatured at a higher temperature. The two strands will separate from each other. After separation of strand, the primers are provided. These primers are basically delineating the region which has to be amplified, a specific fragment of your DNA or a region of your entire genome that has to be amplified. These primers will go and bind to their respective sites, and I hope by this you might be able to grasp it exactly how the primers are binding to the specific target site. I have shown in this cartoon also. Now this is called annealing temperature. This is a particular temperature that depends on the sequence of primer. So normally the annealing temperature is calculated on the basis of the G and C and AT content of your primer. So it is a little bit complex thing, so we won't venture into it more. So the annealing temperature, normally what we keep it at 60 degrees centigrade. So we design a primer in such a way that the annealing won't exceed 60 degree. If at all it goes 60 degree, then it's also not an issue, but generally we keep it at 60 degrees centigrade. And while keeping PCR, the thumb rule is you can keep it at 60 plus minus 5. Either you can keep at 55 or 65, or in between any temperature you can keep your annealing temperature. So finally, after annealing, once your primer anneals, the replication has to initiate. So we call it extension. So extension is done by a polymerase, or DNA polymerase will go and extend the sequence, taking the information from the template strand. It will synthesize the strand which is being synthesized here. So information is provided by your template strand. And DNA polymerase, in this case, it is a little bit different from the DNA polymerase which is present in our cell. I'll discuss in the second slide. But for time being, I just want you to know that primers are the essential part of your PCR. And normally the primer length is kept around 18 to 20 bases. And the most important thing here is that you should know sequence in advance. Without sequence information, you cannot design primer. But then again, you might be thinking that what is the need that if we already have sequence information, then why the need is felt to sequence it again. So for this, I'll give you an example. Say human genome is completely sequenced, but it is sequenced for a reference individual. So if I wanted to check for a gene, say some research had recently been reported that a particular mutation in a gene, it is if people are having that mutation, they are more predisposed to a particular kind of disease. So if I want to check that exact mutation, if that mutation is present in my genome or not, I can take those primer sequences from that particular research. I can isolate my DNA and amplify it from my DNA and then sequence it. So that's why we need these kind of things. So at the end of this extension cycle, we will essentially get two for a single fragment or for an initial single copy, you will get two copies. So you can imagine if you repeat this cycle, say 30 times or 40 times, how many copies you will get from this PCR reaction. So now the thing is about polymerase. So polymerase in this case is because these reactions all these reactions are going at a higher temperature, uh, more than 60, 70, even denaturation we have a 94 degrees centigrade. So at that temperature, your DNA, your polymerase won't be stable. So that's why we need a polymerase which should be thermostable. So fortunately, this thermostable polymerase we have with us now, this was isolated from Thermus aquaticus by Thomas Brock. And this is a very heat stable polymerase. So this is routinely used. We normally call it a TAC polymerase. This polymerase is generally used for your PCR amplification in during your day-to-day lab activities. Apart from this, we have some specific kind of polymerases. Say, for instance, now this COVID testing is going on. So for COVID testing, we go for reverse transcriptase polymerase. It is also a polymerase, but it synthesizes your DNA on mRNA, basically. So there are some specific polymerases, but routinely we go for this particular tag polymerase for amplification of your DNA. Now, now I hope that by this, I will again go back to this your book. In your book, this is given. I tried to explain this in a simple way so that we can understand what exactly is happening inside PCR. Now, second topic is DNA sequencing. So before going into detail of DNA sequencing, I just wanted to give you a brief idea of DNA replication. Again, this cartoon I have made to make you explain how exactly DNA replication goes on. So like PCR, in this case also, and normal in your eukaryotic cells also, for replication, DNA has to unwind first. Then in this case, in replication, we have RNA primer basically, but there's no need to remember RNA primer right now. This is a different story, but just to remember, a primer is needed for replication. And then again, same thing, A has to pair with T and G has to pair with C, thumb rule. So if you have a template strand, you can see at the bottom of your slide, a template strand is there. And in that template strand, if A is there, so in this A, the T has to be, so from this A is basically your information, which is in a template strand. So from this information, if it has to be replicated, this T has to be incorporated in the strand which is being synthesized. So this is how replication goes on. And whatever information is there in a template strand, the corresponding complementary base is incorporated subsequently. Now, this is exactly what is happening during replication, the typical chemical reaction which is happening in during replication. So you have this deoxyadenosine triphosphate. This deoxyadenosine triphosphate is basically a base. In this case, I have given adenosine. Likewise, we have 4 different bases: deoxy, cytidine triphosphate, cytosine triphosphate, guanosine triphosphate, and thymine triphosphate. So I have given you example of DATP. Deoxy, why we call it deoxy? Because at this 2' position, 2' is basically, you can see from this, in this ribose sugar, if we take carbon which is attached to this base as 1, second is 2', and then third one is 3'. I have depicted by arrowhead at 3' hydroxyl group. So we don't have OH anywhere in this ribose sugar except at 3' end in case of your DNA. So it is basically deoxy adenosine triphosphate. Why deoxy? Because oxygen is not, hydroxyl group is not present at 2' position. So this 3' hydroxyl is really very important for replication. So now you shift your attention toward this this double-stranded DNA, how it is replicating. So you can see a primer is bound to this template, and the base which has to now to be incorporated, it is A, because T is present in your template strand. So A has to be incorporated. So how this is incorporated, basically, polymerase is needed. But the most important thing which is needed is your 3' hydroxyl group. So this 3' hydroxyl group from the base, which has already been attached to that particular, or the penultimate base, we can say, or the base which has already been attached to your uh, uh, primer. From this base, this OH is important because this catalyzes the reaction wherein this base, the incoming base will be incorporated. So what will happen? As A has to be incorporated, the OH from the base, which has already been incorporated, will attack on the phosphate of this adenosine triphosphate, and this pyrophosphate will be are released from this. These two phosphates are released, and eventually you can see at the bottom it gets incorporated with the phosphate group. So likewise, this reaction goes on, and the important thing is that you need 3' OH. Now, why I told you that this entire story of 3' OH here? So imagine what will happen if this OH is missing. So if this OH is missing, in this slide, I hope you are able to see the hydroxyl group is not there at 3' position. So we call them dideoxynucleotide triphosphate. These are dideoxynucleotide triphosphate. Why dideoxy? Because at 3' position, hydroxyl is missing. So this is not a normal base. This is different from your DNTP, different from deoxynucleotide triphosphate. This is dideoxy. Both the hydroxyls are missing in this case. So what will happen? Because this hydroxyl is missing in this case. So again, I made a cartoon to explain this. So if, say, at first instance, you can see a C here, and I have put a cross mark here. It means that C is not having that hydroxyl group. So what will happen? If that C gets incorporated in the strand which is being synthesized based on the information of the template strand, so that C has been incorporated now. You can see at the bottom of this picture, the C has been incorporated. But since this OH is missing from this C, so what will happen? No other base will be incorporated. It means that your chain gets terminated here. So that's why we call them dideoxy terminator. Since they are not having that OH group, so this OH won't attack on the phosphate of the incoming base, and so the base won't be incorporated. So we call them terminator. So I hope from these slides you got an idea that what exactly terminator are. So for sequencing, you have to remember this. These are dideoxynucleotide triphosphate. We call them chain terminators also. So this is essentially what is happening here. So now these terminators are having a very important role for DNA sequencing. So to understand DNA sequencing, you have to really focus on it, because it might seem very complex, but when you write it down, all these things on a pen, on a paper, basically, you will exactly understand how the sequencing is going on. So this I have taken from a book, but then again, I have tried to make a cartoon of this so that it becomes easy to catch what exactly is happening. So what we do in sequencing, say in the uppermost, that blue and red, you might be able to see those, that blue and red sequences. This red is basically your primer sequence, and this blue one is your template, which has to be sequenced. So if this sequence has to be this particular sequence, we were interested in to sequence this particular fragment of DNA. So we have to keep 4 different reactions. For simplicity, I have shown 2 here, but in subsequent slide, I'll show you what will happen if you keep 4 different reactions. So you, for time being, you only focus on the first reaction. The first reaction, we have all the bases here. Bases are basically, which I told you, deoxy adenosine triphosphate, cytosine triphosphate, thymine triphosphate, and guanosine triphosphate. They are basically bases, and these are building blocks. These are the bases which are incorporated when the DNA is being synthesized. So in first reaction, all these normal bases are all there, but also we add a small amount of dideoxy terminator. So in the first case, you can see you focus on this first reaction only. What is happening here? We have this, since we have added a dideoxy terminator, and in this case, dideoxy terminator for A only. So it is dideoxy adenosine triphosphate. Likewise, 4 different reactions are there. They are essentially similar except for the dideoxy terminator. So in this case, the dideoxy terminator is dideoxy adenosine terminator. We have cytosine terminator, guanosine terminator, thymine terminator. So now you imagine what will happen when this reaction is being synthesized. So They are, and one thing I want to mention here is that these dideoxy terminators are radio labeled. So why radio labeled? I'll come to this. Radio labeled means basically they are labeled with some molecule that you can easily capture. So, or you can also think that they are labeled with different colors. So in this case, this A is labeled by some color that you can easily capture by some image capturing device. So now you take a pen and a pencil and a paper in your hand, and you try to write down what are the different possibilities here. So in first instance, see if I go and try to synthesize this strand based on the information in template strand. So first base is G. You see uh, the last fragment which I have made in this cartoon. So this 10, why I just for simplicity, I have mentioned it 10, so that 10 means the length of primer is 10 bases. So anything which is added will add on it. So since in this case we have added DDATP, so G will normally be synthesized, chain won't terminate here. So next is T. You see that blue color fragment. Next is T. So T, it means that in the strand that has to be synthesized, A will be incorporated. So you have added dideoxy terminator. So what will happen? That if your chain terminates at this first T that has been incorporated, that the first A. So what will happen? You will get a fragment of 12 bases. G, normally G will be there and A is there. So it is of 12 bases, your chain terminates here. Next possibility is there. You have this G, and suppose if the normal A has been incorporated, because I told you these all four components are already there, we have only added a small amount of dideoxy terminator. So there are chances that normal A will also get incorporated. Say upon first encounter, normal A has been incorporated. So the chain will proceed because chain is not terminated here. Now you can see for A, T will be incorporated. Then again, A is there in template strand. So again, T will be incorporated. Now then you have G, so C will be incorporated. You have C, G will be incorporated. Now again a T is there. So in this, the second possibility is there, your chain will terminate here. So termination is here. So you will get a fragment of 17 bases because you can add these 7 bases to this 10. So the second fragment which you will obtain after the termination is of 17 bases. And finally, likewise, you can also make this final fragment. If there is also a chance that that 17th A, a normal A will get incorporated, the chain will further progress. So because it is not terminated here, so at the end there is again a T. So you can see from, did you see at the top of your slide? Just a minute, I have a pointer. Okay. I hope the pointer is visible now. So this is basically, this is your.
We can see.
Sir, you are able to see the pointer?
Yeah. Yes, yes.
Okay. So now, yeah, this view, I hope now it become easy to understand. So the end base is again T from this template strand. So what will happen? That A will be incorporated here and chain will terminate here. So there are 3 different possibilities in the first reaction where I have added DATP. Now, if I talk about the second reaction, wherein again every other component are same except for dideoxy C. So it means wherever C will be incorporated in your strand that is being synthesized, the chain will terminate. So in this case, you can see. So again, in this blue strand, you can see C is there. So normal G will be there. T is there. Normal A will be there. Then again, AA is there. TT will be there in your strand that is synthesized. Now, this 5th base is G. So it means that C has to be incorporated. So if this C is dideoxy C gets incorporated, then chain will terminate here. So the fragment size will be of 15 bases. Now suppose a normal C gets incorporated. Next G is at your 19th position. So it means that C will be incorporated corresponding to this G. So then your chain will terminate here. So likewise, if you add DDCTP, you will get two fragments of 19 and 15 bases. So now I hope, and now these corresponding bases, this reaction, if I go for electrophoresis, agrosgel electrophoresis, say for this reaction 1, I have 3 bases, and as I mentioned, they are radio labeled. So only the fragments which are having A at their end, they will be visible. So it means all these 3 fragments which are there, you can write A in front of them. Likewise, in reaction 2, if I go for electrophoresis, only the fragments which are having C at their end will be visible. So likewise, you are getting 2 amplicons. You can write C corresponding to these 2 fragments. So this is a simple representation for how your DNA is being sequenced by Sanger sequencing. Now, I told you that if you keep 4 different reactions, anyway, you have to keep 4 only. Otherwise, it won't be possible to decipher the sequence of your target. So the next reaction is, again, all the components are same, except dideoxy is dideoxy TTP. And finally, we have this dideoxy GTP. So now you can easily make that what kind of fragments you will expect in the reaction 3, where you have added this dideoxy terminator. So it will terminate at T. So all the fragments at the end, they will have T, and they will be of different sizes. In the same way, dideoxy GTP also you can imagine, or you can write it down. It is better that after this presentation you try to write it down, because it will become very easy to then it will become very easy for you to understand what exactly is happening in Sanger sequencing. Again, we have these fragments, and I have written the size also. You can count the bases also, and every fragment will be of different sizes because they are terminating at different bases. So in the last reaction, you can see I have 4 fragments. At the bottom, you can see there is a fragment of 11 bases, because the first base in this blue color template is C. So again, G will be incorporated in your synthesis strand that is being synthesized to terminate here. So this is exactly the pattern what you will get. Now, if you run all these things in electrophoresis, so for the 2 reactions I told you, for third one, for DTTP, you get again 3 fragments of different size. I have made these amplicons at different positions because they are of different size. So in electrophoresis, your fragments of in electrophoresis, these are basically of different sizes. So in electrophoresis, your fragments will migrate on the basis of their size. So that's why you will see the size difference also. So for this DTTP, this lane number 3, you can see 3 fragments of the variable sizes. But again, at the end, you will have T. So you can write the corresponding base to this fragment as T. Finally, for this lane number 4, you will have, in this case, we are getting 4 different fragments of variable sizes. So you can write a corresponding base G for that particular fragment. So likewise, if you go on writing the corresponding bases, you will get this first line. You will get this first line of sequence basically, like A, G, T, C, C. Likewise, on the base, these are on the basis of the fragments which you are obtained by after electrophoresis. Now you have to make a complement of these bases. So if you make complement, write T in place of A, C in place of G. Likewise for the entire sequence. So now you can see, you read from the bottom of this line. Now you can see, you check with this blue color sequence, you see that we have deduced the sequence of your fragment C, T, A, A, G, C, T, C, T, C, G, A, C, T. So this is how Sanger sequencing is being done. So I hope I tried to make it very simple, although I do not know how much you were able to understand from this. But if you have any problem, you can write a message in this question answer box also, and you can WhatsApp also. So this is what exactly we do in Sanger sequencing. Now, this is a snippet from your NCERT textbook, but it was not clear. So I mention it here. See, there is a mistake here. In this first reaction, they forgot to add this normal dideoxy ATP also. So it is difficult to understand from this textbook. The reaction will contain all the bases, all the normal bases also, but you have to add a small amount of dideoxy terminator of a particular reaction. You have to make 4 different reactions of this dideoxy bases. So the mistake here is that they forgot to mention the base here, like one dideoxy should also DATP should also be there. So, and then this is again same from your NCERT textbook. I took this picture. So same thing which I depicted in this case. This is essentially the same thing, but I hope from the picture which I have shown here, you might be able to comprehend easily as comparable to the picture shown here. So this is what exactly how Sanger sequencing is being done. Sanger sequencing has, Frederick Sanger was awarded Nobel Prize for this, and it was a landmark discovery because this was before 2005. Sanger sequencing was the only method by which DNA was sequenced, and today, till today also, it is very robust because in this case you can get a read length, means the number of bases that can be sequenced in a one go. So you get a read length of 700 base pairs. Now the sequencers which we have right now with us, they have a very short read length, and the error rate is also very less in Sanger sequencing. Human genome was sequenced by using Sanger sequencing only, but it is a very time-consuming process. It required almost 20 years to sequence human genome by using this Sanger sequencing method. Now, I just wanted to say something about NGS also, because there is a small heading given in NCERT textbook about this next generation sequencer. This is the next generation sequencer, essentially based on the Sanger sequencing method only. The only thing what we have used here is we are using reversible dideoxy terminators. In Sanger sequencing, what we were using was dideoxy terminators. It was not reversible. The moment it gets incorporated in your strand that is being synthesized, your chain was terminated. However, in this case, we are using a reversible dideoxy terminator. So why it is so, I'll tell you. Although I am showing it this in a very simple way, it is not at all like this. It is a very complex chemistry. The instrument itself cost more than crores. This next Illumina, this is basically the concept of Illumina sequencer, which is now ruling this next generation sequencing industry. So this is the simple concept how sequencing is done in Illumina platform, and what is advantage as comparable to Sanger sequencing. I have tried to make it very, very simple. So in this case, we are using 4 different bases, A, T, G, and C. You imagine for time being, they are coded with different colors. A is for green, G is for red, yellow T, and blue C. But they are terminators, but they are reversible. A chemical group is being attached to these particular bases, so that once you remove that group, you can initiate the chain from that point onwards. So you see this example. I'll point it here. So it is ATGCT. This is your primer. So in place in the strand that is being synthesized, taking information from a template strand, T will be incorporated. So T is yellow in color. So that image capturing machines are there. They will immediately capture the color. So if they capture a yellow color, it means that the sequence is T. But the reversible group is very important here, because if the reversible group is not attached, you have to keep a separate reaction. So imagine what if because the chain terminates here, there is no free OH here. So imagine what if we remove the reversible group. So after image capturing, once we capture the image, we get an information, okay, the base is T. So we can remove the group. So to remove the group, there is a separate chemistry. The terminator group is removed. And again, the reaction goes, the reaction is repeated again. So what will happen? Now T is there. So in your strand that is being synthesized, your A will be incorporated. So A is green in color, image will be captured, machine will immediately say the base is A. Again, the group will be removed, and again, this process will go on like this. So it is exceedingly simple to understand, but it is not at all like this. It is really very complex mechanism. But the principle is this only. You have a reversible terminator, and you have very robust image capturing device, and on the basis of those images captured. Now, the important thing is that a question might be, you might be wondering like how to sequence the entire genome. So sequencing a fragment of DNA is a very simple process. You can sequence in a single day. See, I told you that in Sanger sequencing, the read length is 700 bases, and this Illumina sequencing, which is the gold standard for sequencing nowadays, the principle uh, which I have shown here. So in this case, the read length is very small of 100 bases, and human genome is of 3000 bases. Now 3000 mega bases. So now you imagine how many small fragments, if you go on sequencing a human genome based on this sequencer. So it means that you have to break your genome first and then sequence your genome. You will get small bases, 100 bases, you will get billions and billions of bases. So how to align those bases is a real challenge. Now, just to give an idea what exactly the problem is. So I hope you know who she is. So if I make a jigsaw puzzle out of this, and if I break this picture into, say in this case, there are 8 different pieces. So, and if I tell you to align all these pieces again and to make a complete picture, it becomes very easy for you because they are very big in size. You can easily fit those gaps and then you can make a complete Anushka Sharma. But you imagine if you break these pieces into thousands and break this picture into thousands and thousands of pieces. So this you cannot do by your own. You need high performance computers. You need supercomputers, which will align those pieces and then finally make a complete picture. This is exactly what happens when we sequence our genome. We break our DNA into small pieces. These small pieces are sequenced by the Illumina sequencer or Sanger sequencer, but nowadays we go for Illumina sequencer also. Although we have some robust platforms apart from Illumina sequencer, but Illumina is so far the gold standard because of the less error rate. So you have this to break your DNA into small pieces. Now these individual pieces are sequenced. Now after sequencing, the real challenge is to align all those pieces. So alignment means, I told you, this is again the same color coding. Machine will give you in a color coded form. Alignment means you have to align those pieces on the basis of overlapping information. These pieces are not exactly like cutting something. You are sharing your DNA, and it is randomly shared. There might be pieces which are having good overlap, and based on that overlap only, these pieces are aligned. So in this case, you can see the alignment is there, and finally on the basis of alignment, a complete sequence is being made. But this is not at all an easy job. It is really a very daunting task. Basically, you require people from diverse background. You require people from computational biology. You require people from mathematics, statistics to make a complete genome. So that's why now biology is becoming interdisciplinary. The people from diverse background are coming to this. And now you imagine the situation of COVID right now, what we have. We are midst of this ongoing crisis. So, and in this also, this Illumina and all those things, all this sequencing platform are really doing wonders. Recently, I came across a machine, Illumina chemistry, where they are able to multiplex your testing. Like they can pool your DNA from different individuals, and they can sequence in one pool. Then after they can deconvolute those pool basis on the basis of some barcodes, and this will dramatically reduce the testing cost. So this is how the technology is going. Now finally, after sequencing, the most important thing, it is mentioned in the NCERT textbook also, and I was given this task also to make you understand what exactly annotation is. So before going into detail of annotation, I just want to seek your attention on this particular thing. Now, this is basically a structure of a eukaryotic gene. The structure means a gene has some specific signature sequences or some features, basically what we can say. A gene should have a promoter. Promoter is a DNA sequence that guides the expression basically. If promoter sequence is not there, the expression won't be there. So these are some, it is also, when you see for a gene in a particular fragment, so you check for promoter region, then a gene should contain exon. Then it should have introns also. Exons are basically which codes for your protein. Introns are intervening sequences. They are normally spliced from your DNA while the formation of mature RNA. Then finally, there might be multiple exons. There might be multiple introns. You have a start codon. Start codon. Normally, start codon is ATG. I hope all of you know from your textbook knowledge. ATG is your start codon for eukaryotic genes. You have your stop codons also. UAA. Wait a second. I just. You have stop codons also, like UAA and this, and there are two more. So. Then you have Terminator sequence also that terminates your transcription. So these are some features of your gene. On the base, if a particular fragment of DNA have these kind of features, so you can say that it might contain a gene, but you cannot say with certainty that it might have a function, that it cannot assign a function by seeing the structural features of a DNA fragment. For assigning features, there are different methods. For time being, I just wanted to let you know that gene has some specific features. And whenever you are provided with a sequence of DNA, you have to check for features first. Whether the piece of DNA is having these features or not, then only you will proceed further. So I've written it here. You have an unknown sequence. Then on the basis of that unknown sequence, you can have to predict your gene. For prediction of your gene, you cannot do it manually. Again, you require computer platforms. Those are platforms are you require softwares. Those softwares are fed with some information, and on the basis of those information, they go and predict these features in your unknown sequence. Then finally, functional annotation. Functional annotation can be done. There are two ways how you can annotate a function to a gene. Annotation means basically you are providing a function to a piece of a DNA. So the one way is basically, say, suppose I isolate a fragment of DNA from myself, and I know that it is for me it is an unknown piece of DNA, a sequence, unknown sequence, and I'll go for a BLAST search. So BLAST is a database basically where the information are being submitted. Anybody, if uh, if at all someone has worked earlier on the piece of DNA which I am working, so he might have annotated it, he might have done the functionalization also. So they submit these kind of information in your BLAST database. Say, so for if I have an unknown piece of DNA, I'll directly go for a BLAST search first. I'll see whether it has already been annotated or not. If annotated, well and good, I can directly say that this piece of DNA is having this particular function because in BLAST search it was given like that. But if it is not there, the only way how you can annotate a gene is to transfer that gene to an individual which is lacking that gene. Say, if I say I have two mice, one mice is deaf, the mice cannot hear, but I'm working on some genes which are related to hearing, and I landed to a piece of DNA from a mice which was able to hear, and I took that piece of DNA, though it was unknown to me, but I transferred it to a mice which was deaf. And fortunately, what happened? Now the mice which was deaf, he was able to hear now. So it means that the gene is somehow related to this mechanism of hearing. So I can say that gene is responsible for hearing. And in fact, this was the first gene which was cloned by this way, the gene for hearing in case of mice. So you need a mutant in this case, which is lacking for the particular piece of DNA. If you complement that mutant with the piece of DNA which is with you, and if the function is restored, it means that the piece of DNA code for that particular protein or particular enzyme which is being involved in that particular pathway. So just to give you a flair of what exactly annotation is, you can see if you have a piece of DNA. So if you see it, what exactly you can infer from this? I hope you cannot infer anything from this because this is nothing but letters like A, T, G, and C. So what to do with this? So you cannot do anything with this. Even if you have sequence information also, it is of no use for you. It is only just like ATGC, ATGC. So this is, I told you, if I put this sequence, I feed this sequence to a particular software. Softwares are basically made on the base of our knowledge. So if a software is fed with some important parameters, that I train my software that, okay, you check this parameter in your piece of DNA. So in this case, I did it. This is for this sequence only, what I'm showing it here. I did intentionally for this class only. So I fed this sequence to this particular software. This is FGenish as a software for gene prediction. So on the basis of this, the software has predicted 2 genes for me. This, just wait a minute, I just go for point. This is first one, this is second one. And normally what we do, we go with the longer one. So, so this is the case. The software did predicted these 2 genes for me on the basis of these sequences like stop codon, start codon, all those things. And finally, it gave me 2 sequences. Software said that these are the 2 sequences that might be the genes, but software is not giving you any idea that they are gene or not, but it is giving a possibility because they are having those signature sequences. So I'll go with the longer one. You can see the software on the basis of this ATG and this CTT here. CTT is basically UAA, stop codon, ATG is start codon. So this is basically what software gave me, a complete sequence from ATG to CTT. Now, still you don't know the function of this gene. So I told you, you have to go for a blast search. If somebody has already worked on this particular gene, so they might have annotated on the base of it, you can easily get the function. But if nobody has worked, then the only way left with you is you have to annotate it. And I told you, for annotation, you need mutant. Without mutants, you cannot annotate. You have to complement the mutant phenotype from the gene of interest. Then only you can annotate. And if the mutant phenotype is being complemented, then you can easily say that this gene is over. This gene codes for this particular protein, and this protein is being involved in this particular phenotype. Phenotype is basically the appearance or some particular trait, I can say. So this is how it goes. So this is the final last topic of mine. Hybridization technique. I hope so far I am able to convey what exactly the task was given to me. So now the final thing is this hybridization technique. So now the question, my first thing is that why we need hybridization? There are different methods in molecular biology, but to learn those methods, first we have to ask question that why exactly we need all those things? Why exactly we need southern hybridization? Why exactly we need DNA amplification? So I told you why we need PCR, because we want to amplify. Why we need Sanger sequencing, because we wanted to know the sequence of DNA. Why we need this annotation, because we wanted to know what exactly that gene code, what exactly that piece of DNA is doing in your body, or what exactly that piece of DNA codes for. So that's why these are all stepwise thing. Now this is given in your book, hybridization technique. So why we need hybridization is basically, now hybridization, we normally do hybridization in the present day for different things. Normally in case of we go for copy number estimation and all those things. But initially hybridization was a very important method. Why it is so? I'll give it, you imagine a situation before 1980s when PCR was not there. Say I isolated a piece of, because these model systems means those which are very well studied. So I'll take an example of mice or guinea pig. So say for instance, if you have sequence information of mice, because people used to work on mice a lot, and if you wanted to check that whether that sequence information means you can have a sequence for a specific fragment. Now we wanted to check that whether that sequence is present in human beings or not. So now it is easy for you. What you can do, you can design primers and you can go and easily amplify the primer in your human genome. But before 1980s, it was not like that. So what was done at that time, this, you can take that piece of DNA, you can label that piece of DNA with some color. I have shown here with a heart of green color. So if this piece of DNA is labeled with a green color dye, and this black is basically your human genome, and I wanted to see whether this piece of DNA or the sequence, at least some similar sequence, because see, the genes which are important in your say I'll tell an example. example of, say, glycolysis. Glycolysis is basically an important pathway of sugar metabolism. So it is conserved across genera, across species. So the genes which are involved in glycolysis in mice also, they will be present in human beings also. There might be some sequence divergence, but if you check for similarity, largely they will be similar. So if I take an example, same that glycolysis gene, I take a gene from this mice, and I labeled this gene with some particular dye, and then this human genome is there. I'll go for hybridization. So this fragment will only hybridize to the region wherever it will find complementarity. Otherwise, if the region is not there, it won't hybridize. So it will go and hybridize. And if because it is hybridizing there, and I have attached it with a fluorescent molecule or something, say a color. So if I get a color, it means that a sequence which is similar to that particular fragment is present there. So this is basically why we do hybridization. Now the thing is there, why we go for blotting, southern blotting. See, in first instance, I took this black is your, just a second. This is your human, and this is for the piece of fragment from mice. If I go and directly hybridize this fragment with this, so because I have to go for electrophoresis first. So what will happen now? Say this is the fragment. So because I haven't chopped my DNA into small pieces, I'll get a single band in electrophoresis. So what will happen after hybridization also, you will get a single band. So this is very less informative. You don't have any information from this. Only thing you get is that, okay, the fragment is being present. But what if I break this DNA into small pieces? If I break my DNA into small pieces, and then I go for electrophoresis, after electrophoresis, they will be separated in a particular media, say an agarose gel, and this gel I will use for hybridization. So what now it becomes informative, because in this case you are getting the size also. What is the exact size? Because if you run a marker, a ladder here, marker means our DNA fragments for which the size are known. So if you run this marker here, so in this case, so by this you can easily infer the size also. So it becomes very informative. So in both conditions, in both situations can be followed. But if you go by this way, so it becomes the second way, it becomes very informative. So what we do, so this was just a cartoon to explain, but again it is not so simple. The first thing is that what we do, we break our DNA into small pieces. Then we go for an electrophoresis, separating those DNA in a media. Then we go for hybridization of this fragment to your to these fragments which are there in your media. And then finally, if at all a complementary sequence or the sequence which is highly similar to this mice piece of DNA, if it is there in human genome, you will get a signal. So then in this way, you can say, okay, this is also present in humans also. So this is why we go for hybridization. Now, again, I told you it is not so simple. So what is done exactly is you cut your DNA with enzymes. Enzymes cut your DNA into small pieces. Then after we go for electrophoresis. Now electrophoresis, we run your in electrophoresis, the DNA is basically separated in a particular media, agarose. So that is not easy to handle. So it is desirable to transfer the DNA fragments from this agarose gel to a membrane, because membrane is nothing but a paper kind of thing. So if something is there in paper, so your DNA is now transferred to those paper. So in hybrid, what we are doing in blotting, basically, we are transferring your DNA, the fragments after electrophoresis, which you have obtained after electrophoresis, to membrane. So for transferring, again, this is nothing but your capillary phenomena that is given in your 8th standard books also. By this capillary phenomena, the sponge, a buffer tank is there. On that buffer tank, the sponge, 2 or 3 layers of sponge are placed. After that sponge, your gel, agarose gel, in which you are having your fragments that are being separated, by electrophoresis. You put that gel on above those sponge, then a membrane is there. Then again, some filters are there, paper towel is there. Finally, you put a weight over this. So what will happen as because of capillary, the buffer will start migrating, and due to the migration of buffer, your fragment of DNA will also start migrating upward. And because at the top of your gel, you have placed a DNA binding membrane or a filter or a paper, you can say. A paper is there, DNA binding paper is there. So your DNA will migrate from your agarose gel to those paper. It will bind to that paper. So finally, and now that paper you can use for this hybridization. So what will you have this uh, what is it? You have this fragment of this mice fragment is there with you that is labeled. You can go for hybridization. And so this, in this case, we call this fragment as probe. So probes are nothing but your DNA fragment, which are radio labeled or labeled with any other molecule that can easily be captured by some image capturing devices. Radio labeled because it is easy. You can easily capture by X-ray film and all those things. Otherwise, we have now very good labeling molecules that can be attached to your fragment of DNA. So this is I capture. I think I tried to whatever it is given in your NCRT book. I tried to mention it here in simple terms. I hope that you might be able to catch it. So now sir told me to put some questions here, but unfortunately, the only question is here. The heading is here. Questions are not here because that slide I left in my office computer. So anyway, sir will give you question by WhatsApp or in this chat box. He can share some questions. So I thank you for your patience hearing, and please feel free to contact if you need any kind of help or any kind of clarification related to my lecture. Thank you once again. I am over, sir. I'm over.
Okay. Okay. Thank you, Deepak. There are a couple of questions which I will read out, which you can answer. I'll read out one by one. Are you able to hear me?
Yes, sir. I am able to hear you.
Okay. The last questions come first because you had just talked about Southern.
Yes, sir.
How DNA is denatured during Southern? One question. And related one, when does double-stranded DNA become single-stranded in the Southern? So this is concerning the Southern.
Okay. So for denaturation, we use this one normal HCl. So by this, your DNA denatures, and then it becomes single-stranded. So, sir, is it what?
Yeah. Because see, denaturation happens in PCR.
Yes, sir.
Temperature. Here it is chemical. Okay.
Yes,
so
sir.
That's what I think they wanted. How at what stage
Stage,
it is denatured?
during before transferring to this membrane. So once you have your DNA in this agarose gel, so because if you make your first thing I told you in my presentation also, the answer is why we at all we want to make it single stranded. So the thing here is that if it is single stranded, so it becomes easy for the transfer of your DNA from gel to your membrane. And because single stranded, you need hybridization also. Probe has to hybridize on single stranded DNA. You have to open your DNA first for hybridization.
You want to highlight being academicians and all the academicians that even we are conducting.
Sir, is want to ask something?
No, somebody else is speaking. I do not know.
Okay. Okay. So single stranded means because your probe has to bind. So that's why we have to make it single stranded. And in this case, we go for, because we cannot keep your agarose gel in some oven of 94 degrees centigrade, because anyway it will melt. So that's why you have to use chemical method denaturation. In this case, we use 1 normal HCl for denaturation. 0.1 normal HCl, sorry. 0.1 normal HCl.
Okay. Related in the sense I had mentioned, perhaps you skipped, because only we wanted all kinds of blots, northern, southern, and western. Okay. So just briefly tell the difference between southern northern, and western. Very briefly, I don't think we can take it. Maybe we'll take if they want detail on Saturday. But at this moment,
I'll
you can
make up
just.
a separate presentation for it, only for hybridization dot set. But for time being, I'll tell you.
Yeah, just mention.
In Southern, what we do basically, Southern is basically your DNA hybridization. So your probe is your DNA, your fragments are also your DNA. In Northern, what basically we are doing it is RNA DNA hybridization. Your probe is DNA, but your the fragments which are on your gel are your RNA fragments. And in Western, it is your protein DNA hybridization. So all the methods are essentially similar, except what you want to check. Northern is basically for your expression. If you wanted to see whether a particular fragment is being expressed or not in the sample which you are considering, so you go for Northern. If you wanted to see whether the protein is being present or not, then we go for Western hybridization. Western blotting, basically, we say.
Okay. The hybridization maybe we can take up separately. This is in short. That's fine. There are a couple of other questions that I will ask again. If we are cloning, this is a question which might be in the minds of many. If you are ultimately cloning in bacteria,
Yes, sir.
then why are you doing PCR? This is the question. In the sense, so without PCR, can you directly clone? In other words, and what are the issues if you want to directly clone from the genomic DNA?
Yes, sir. I'll answer that question. So PCR, I told you for PCR, what we are doing is basically we're making multiple copies of a small fragment. So without PCR, you can clone only if you have multiple copies of that fragment. This is the only way, basically. So when I say if I wanted to clone a specific fragment of DNA, so
Hello, you are not audible.
sir, am I audible, sir? Am I audible?
Yeah, no.
Okay. So in PCR, basically, if say for instance, I want to clone a gene X from human being. So first is that first thing is how will you identify that gene X? How will you isolate that gene X from human being? So for isolation also we go for PCR. I hope I am able to convey it.
Uh, maybe I will slightly clarify.
Okay.
See, if we do not know anything about a gene, let us say, let us take hemophilia gene. Before it was discovered, there was no way that I could clone. All that I could do is make a pieces of this one, do a standard cloning, and then from the whole lot of recombinants, somehow I had to identify that particular piece which contained the gene of interest. So that's a very, very long and tedious process. So once we know the gene, it is far to clone it. Why we want to do PCR? Because we want to have more number of copies first. Because if you have only one copy of a fragment, it will be very difficult because everything is probability based. The recombinations, the ligations, picking it up into the bacterium transformation. So we need more copies. That is why basically we take a piece of DNA, amplify it by PCR, and then clone it. And it is also that we want to use this gene with a different promoter. So that is why we would like to do a PCR and then clone it. I think that should explain it.
But sir,
Yes.
can I just chip in?
Yeah.
I thought the question is, if you have cloning, why have PCR? So in the compared to the morning session where cloning was described,
Yes.
the difference is the amount of DNA. In the earlier morning classes, when she talked of she can get it amplified into a bacterial plasmid or whatever vectors. In this case, the advantage is that even if it is at a very low number, amount is small, you can still amplify it. So PCR gives you, in that sense, a very low amount which can be amplified. In the earlier case, if you had to clone, you should be able to visualize the band and cut it and insert it. So you need a stoichiometric amount in those cases. In this case, you are able to amplify it. So there is a subtle difference here again, in contrast to bacterial amplification to the enzyme amplification. Without getting into a vector, you are able to make multiple copies. I hope I make sense.
Yeah. Another question many people have asked: why primer is needed for replication?
Yes, sir.
Why primer is needed for replication?
Primer is needed for actually DNA polymerase cannot synthesize a strand on a native strand. मतलब अपने आप कुछ नहीं कर सकता सर.
De novo नहीं. हाँ, that's what. It always needs something to begin with.
Actually, RNA polymerase. That's why this is a very important question, sir. Actually,
Yes.
that's why in this case of replication, in case of normal cell, अभी तो हम DNA primer कह रहे हैं, वो RNA primer होता है. Because RNA polymerase is primase is basically which synthesize that RNA primer in the normal reaction which is happening inside the cell. So primase जो होता है, it is nothing but an RNA polymerase. But RNA polymerase का property होता है, वो DNA के strand से normally synthesize कर सकता है without any primer. But DNA polymerase don't have this property. It need at least that OH is needed to synthesize that or a new strand on based on the template strand.
Okay. There is another question. They want you to explain again how these overlapping sequences are aligned.
Sir, वो अनुष्का शर्मा मैंने दिखाया एक्चुअली अनुष्का शर्मा का सर जो दिखाने का रीजन ये था, बट इसमें एक प्रॉब्लम था, इसमें ओवरलैपिंग नहीं था. अदरवाइज this question is very genuine here basically. So alignment is if we make means if they try by writing a sequence in a paper and make the overlap of those sequences, then they can easily understand how overlapping sequences are being aligned. So in this case, I hope this slide is visible. So if I align this sequence on the basis of the color of these dots, so you can see these yellow and this violet dots are common in both the fragments. So on the basis of these common dots, common in this case, in the second one, in this second one, these dots are common. So on the basis of this common, if I change this dot to bases, so it means that these bases are common. So on the basis of this common sequence, they are aligned. I think the question here should be why we get overlapping sequences. So
no. What we can do is we can revisit this question on Saturday. We'll note it down because it can be explained in a simpler way. We'll do that for Saturday. Perhaps the last question for that I can state now is how is PCR used in evolutionary studies?
Okay. See, PCR as such is not used for evolutionary studies. PCR is basically what we can do. Say, for instance, I want to study human evolution. So for human evolution, some specific fragments of some specific sequences are known, which are evolving very rapidly. So if I know a sequence, I design a primer. So I can take DNA from different population, from different human establishments. Like I can take a Hispanic population, South African population, people from Andaman and Nicobar, Lakshadweep, and then amplify that particular region, which is being known to be highly variable from all these populations. So that PCR will do this. But for going for evolutionary studies, we have to then again see for the divergence between those sequences, how diverged they are or how converged they are. So, but as such, PCR is not used for evolutionary studies. It's only a part of.
Yeah. by amplifying, you can compare and do every evolutionary studies, basically.
There is only a machine to amplify DNA, nothing else.
Yes. Many people have asked this question. How much time it takes for PCR and RT-PCR? Because I think all these questions are coming because of this COVID. You can throw some light upon, in the sense, what are the actual time for the PCR or RT-PCR and the overall time that people, take to give the results of a COVID or anything.
Okay, sir. So in case of PCR, so PCR is very simple. I'll tell you why. Because we are working on DNA. So when we work on DNA is very stable molecule. It won't get denatured very easily. So in PCR, I will skip this primer designing part, because once you have primer in your hand, so in PCR to get result, it won't take more than 3 hours. Why it is so? Because now we have machines, PCR machines, which are of very good ramp speed. So you can complete a normal PCR, say if you are amplifying, and it depends on the fragment of DNA also, which you are amplifying. Say I'm telling this for a fragment of DNA, which is a 1 kb in size. So if you are interested in a fragment of DNA that you wish to amplify is of 1 kb in size. So normally to make 2 to the power 35 copies, 2 to the power 35 is basically why I'm saying, because after every cycle you will get 2 copies. So after 35 cycles, you will get 2 to the power 35 copies. So to get 2 to the power 35 copies of that particular fragment in PCR, by skipping the primer designing and ordering time, it will take not more than 3 hours, and you can take half an hour for gel electrophoresis also. So in 3.5 hours, you can easily get the results from your PCR reaction. However, for RT-PCR, it is a time-consuming job. Again, in this case, probes and RT-PCR is a little bit complex, because in this case, different chemistries are used. So I'll take an example of TACMAN probe. Although in lab we use cybergreen, but for COVID testing, TACMAN is being used, because the error rate for TACMAN are less, but the cost is more. So normally in COVID also we have 2 primer only, but apart from those 2 primer, we have this TACMAN probe also. Probe is labeled with some molecule. So if I keep aside the cost of, the time of designing primer, ordering primer, synthesizing primer. So in RT-PCR, the most important thing is isolation of RNA. So isolation of RNA is itself a very complex process, or a very tedious job, because RNA is very unstable, because RNA is normally degraded by RNAs, and RNA is a very like it is present in everywhere, in your tears, in your sweat. So while isolating RNA, there are fair chances that RNA might get denatured. So the most important task here is to isolate a good quality of RNA. The major challenge also is to isolate RNA. Once you have RNA, there are several standardization steps. So if you skip those standardization step also, because it will take more than a week to standardize. But once you have standardized everything in a lab and you have RNA with you, so for RT-PCR, it will take not more than like say 1 hour 45 minutes or 1 hour and 30 minutes. And it is RT-PCR is, if you talk the duration in which you get result, if you skip all these things, DNA isolation, primer and standardization. So RT-PCR is faster as comparable to your normal PCR, because in RT-PCR you can easily see those things in a real-time fashion. A peak is there in computer. In RT-PCR, there is no need to go for electrophoresis. You can easily see the peaks in your machine or your software. So the time duration for RT-PCR as a machine per se is less as compared to your PCR, but the process is really very cumbersome for RT-PCR as comparable to your conventional PCR.
Okay, I think we have covered most of the questions, but there are many other questions which we can take on Saturday.
Yes, sir.
Because we will have to collect them. I have tried to answer some of them, which are short. We will take up the rest of the questions. Anyway, we are downloading everything. Wait till Saturday for more questions to be answered. I think with this, we close this afternoon session. And thank Dr. Deepak for an excellent presentation, although we had